Crispr-Cas9, bring me a gene, encoding for a specific protein. Make a few snips at this coded locus. You work so well inside a streptococcus. Cas9, I'm so alone without your scissors in my chromosome. Cut me up and do it clear. CRISPR-Cas9, bring me a gene.
CRISPR was first discovered in bacteria. Bacteriophages fight against bacteria. They do this by attaching and injecting their viral DNA into the bacteria and hijacking the cell's replication machinery. Most of the time, the cell will die. But if the bacteria manages to fight the infectious virus, it will then save a piece of their DNA. This is similar to memory T cells, but in the bacteria, it saves it into CRISPR. So if the virus attacks again, the bacteria makes an RNA copy for a protein called Cas9, who will then search for that same sequence. It will compare every piece of DNA to the sample piece of DNA. When it finally finds a perfect match, it will then cut the DNA, protecting it from the virus. However, the true magic happened when scientists figured out that CRISPR is actually programmable and can be used for not only viral sequences, but can target any sequence. All you have to do is change the guide RNA sequence to match whatever sequence you want it to edit. When CRISPR reaches the nucleus of a cell, it will find and attach to the matching sequence. It'll make sure that the template is a 100% match. It will then use mini molecular scissors and cut the DNA. Your cells will then sense this DNA damage, and it will try to repair it. And since our repair systems have high error rates, this most likely results in a mutation. However, instead of this mutation occurring, scientists can add a piece of DNA. The DNA will carry the sequence of whatever they wanted to add. It will then ligate to the cut end, and then you get a brand new piece of DNA. This idea was then tested in HIV-positive mice. They were injected with CRISPR and found to have a 50% decrease in infectious cells. It's predicted that in the near future, CRISPR will cure HIV along with many other viruses like HPV. Ultimately, CRISPR is in testing to eventually eliminate cancer. Cancer cells replicate unstoppably, and our immune systems have a hard time detecting them to fight against them. But CRISPR could edit our immune cells so they can fight better. There are also genetic diseases that range from mild to deadly. But most genetic diseases are caused by just one incorrect nucleotide. So we could use Cas9 to change these single nucleotides. It has the possibility to cure many other genetic diseases. However, these changes will die with you, unless they are made in reproductive or embryo cells. And that brought us to genetically modified humans.